In this lesson, we are going to see how this enormous task of human genome project, where the entire genetic composition, the genome, had to be coded, was done. So, we are going to deal with the methodologies adopted towards human genome project, what they were and how it was done. There are two methodologies, one is EST and the second one is sequence annotation. Sequence. Now, what is EST? We are going to see the first methodology of coding the genome of humans. It is expressed sequence tags. Now, the name is showing us little bit of, giving us a little bit of idea what it would be. That the genome has certain sequences which are expressed. That means the genes, the expressed part of the genome is sequenced using RNA. So, whatever part of genome is making the RNA, using that RNA, it can be deciphered what the genetic composition for that would be. That means what the DNA sequences, nucleotide sequences for the given RNA would be. It is very simple, as we did earlier, that if you have an RNA being formed, you can find out what would be the coding strand and what would be the template strand of the structural gene which is responsible for making that particular RNA. So, it is quite clear to make it out that what would be the sequence responsible for production of that particular RNA. So, that methodology is known as expressed sequence tags, where you are targeting only those sequences of DNA which have expressed themselves in the form of RNA. So, you are going to use the RNA that is produced out of the DNA. That means you are going to target only the genes which are expressing themselves in the form of RNA. From that RNA, it could be made out what would be the sequence of the genome. Okay. The second methodology is sequence annotation. Now, this methodology aims at coding the entire genome, whether expressed or not expressed. That means all the 3.3 billion base pairs would be found out what they are, they would be noted down, the nucleotide sequences, what the base pair sequences which would be present for the entire genome, irrespective of the fact which is being expressed, which is not being expressed. So, that methodology is sequence annotation. Now, we are going to aim at this and see what actually this methodology is all about. In sequence annotation, you know that this entire genome would be a long thread like structure, the DNA present on the chromosomes. If we are talking about 3.3 billion base pairs, it would be as long as 1.5 meters, something like that approximately. So, that much length of DNA has to be coded. Now, how it would be coded? It's not easy to code the entire length, you know, we talked about the complexities because it is enzyme involving process. So, the entire length in one go is not possible. So, we cut small sequences. Now, again I would repeat it, irrespective of the fact whether it is being expressed or not expressed, the sequences would be cut into small fragments, then they would be cloned in specific vectors which are bacterial artificial chromosome and yeast artificial chromosome, otherwise abbreviated as BAC and YAC. We are talking about sequence annotation over here. So, in that we have BAC, that is bacterial artificial chromosome. Now, my B looks like a symbol, but it is simple alphabetic B. Now, we have yeast artificial chromosome. So, we use simple cells, we put a vector which has the specific sequence cut out of the genome, we clone it, we get the DNA sequence amplified, and then we figure out that what is to be annotated out of that. Okay, that sequence is annotated. Now, the these two artificial chromosomes are used where the sequence would be amplified using the vectors. Now, there would be DNA sequencers. There is an approach of DNA sequencing. We use artificial DNA sequences to find out what that particular sequence is coding for, or what that particular sequence is being noted for. So, these DNA sequencers, this approach was given by Frederick Sanger. He said that it is better to use DNA sequencers. He gave us these sequences, and he is the one who is responsible for sequencing various amino acids. When we talked about genetic code, we get to know that this person gave us many amino acid sequences, what glycine is made for. You remember that chart where we talked about various amino acids and the genetic code for that. So, he is the person who is responsible for giving us that huge amount of information in that. So, he said that it is better to put those amplified sequences into DNA sequencer, and from that we can extract out the information that we are seeking, the DNA sequences. Annotation could be done. Now, these DNA sequencers, they used the methodology where the overlapping sequences, the overlapping part of the segments of DNA was taken into consideration. Now, the overlapping part is so huge that you have to depend on computers for that particular methodology. You cannot do it in it is not in the potential of human mind to keep record of such a huge, you know, overlapping sequences. So, they used computers, and it took a long time to find out the sequences, and later on they were annotated, and this is what sequence annotation is all about. Also, there is another methodology in this sequence annotation only, that they use the polymorphism for the overlapping sequences which are present in the sequence annotation method. The polymorphism that is shown by restriction endonucleases. Now, the restriction endonucleases are certain enzymes which cut DNA at specific sites. Now, they are highly specific. That means, if a restriction endonuclease is showing polymorphism, that it is cutting at many sites, we ought to have some sort of overlapping. I am going to repeat it. Restriction endonucleases are the enzymes which cut DNA at specific sites. Their specificity is of great degree. That means they are highly specific. They are going to cut at specific sites. For that, they need a sequence. Now, specificity, if it is a nuclease, it is going to be in terms of a sequence. Now, if that sequence is getting repeated, that means if the nuclease is cutting at many sites, it is finding that sequence time and again. So, you get to know that there is lot of polymorphism in the sequence, and that shows overlapping. And from that polymorphism, we can get an idea what the sequence is all about. And all this comes under this methodology that is known as sequence annotation. So, we have two methodologies of Human Genome Project. One is express sequence tag, rather simple one, and this is a long process where we have to depend on the memory of computers, their technical superiority over the human brain in terms of memory storage, and these methods which have been given by certain scientists like Sanger, and our knowledge from genetics that we have gained earlier while coming to this stage that we could code for Human Genome Project. This is all being utilized in this methodology. So, I hope you are clear how this enormous task, I am saying it again, how this mammoth task has been carried out and has made us to reach at a stage where we have certain salient features of this entire project, and those salient features are featured in the next video.